I'm a geneticist. I'm a professor of genetics. I work here at Griffith University on the Gold Coast, and I want to give you a bit of an overview on some research that's related to trying to develop better diagnostics and new treatments for diseases, and that is trying to personalise medicine, to actually give you some medicine based on your genetic profile. So I work at the Griffith Health Institute. In fact, I'm the director of that. This is an institute of health and medical research, primarily located here at the Gold Coast. We have over 500 staff and research higher degree students, and we undertake research in a variety of biomedical and clinical areas: cancer research, cardiovascular research, neurosciences, mental health, population health, infectious disease, and also genetics or genomics research. So I have been involved with the Genomics Research Centre for a number of years now, and the aim of all of our research is try to identify genes involved in common human disorders. Why? Well, basically we do that so that we can work out what's going wrong with the gene. We can use that information to diagnose people who might have disorders, and we can also use the information to develop new forms of treatment. And we work on various disorders ranging from multiple sclerosis, cardiovascular disease, osteoporosis, neurodegeneration, right through to various types of cancer. Now, I'm sure everybody is aware of the Human Genome Project, and I suppose the Human Genome Era, or the Genome Era, probably started with the release of the first human genome, which had been fully sequenced, which was released in April 2003. This was just a little bit shy of 50 years on from when Watson and Crick first came out with the structure of DNA, but it was a big 50 years to go from defining what was DNA, what was the structure of it and how it replicated, to actually being able to sequence a whole human genome. So the big question is, what's going to happen in the next 50 years? How are we going to use that information? And how are we going to use the information that's now coming up from many more genomes, not just the original one person that was sequenced? So there'll be a lot of changes. And in terms of how the information about these genes relates to disease, well, we now know that there's well over 1,000 human genes that have been identified as playing an absolute causal role in disease. But there are many more gene variants that have been associated with susceptibility to various disorders as well. Most of the genes that are truly involved in causing disease tend to be single gene disorder, meaning that if you have a mutation in this gene, you will get this particular disease. But in actual fact, it could be that there are many genes that interplay together to cause disease. And so we're going to have to start looking at putting information together when we're starting to define what kind of disorder you're prone to and what sort of treatment can be used to actually get over that disorder. Now, the information we have at the moment and the information that will happen in the future will relate to diagnosis, better forms of diagnosis, like we currently do DNA testing for things like cystic fibrosis or Huntington's disease. But it'll also be important in developing new sorts of therapeutics, such as information about a particular gene that was implicated a few years ago in chronic myeloid leukemia, and a treatment that's been targeted to that gene and found to be very successful in those patients. So the term pharmacogenetics is probably an area that's really increasing in terms of the sorts of studies that are happening at the moment. And really this is the study of genes and how they relate to different disorders, and in particular how they relate to treatment of disorders. So how do genes relate to drug absorption, distribution, metabolism, targeting, but also safety and efficacy? How safe is the treatment in terms of genetics, and also how useful is it in treating your particular gene problem? Now, you can undertake the sorts of studies to identify what genes are involved in these different types of drug treatments by looking at DNA, by looking at the product of genes, which is RNA, or by looking at the proteins that we can find circulating. And I must say, this is a real growth area at the moment, and a lot of the studies in pharmacogenetics are really just starting. But basically what they're all about is trying to tailor an appropriate treatment to a particular genetic problem in an individual. And so it's really leading to personalised medicine. And what are the benefits of personalised medicine? What's important about it? Well, basically, if we can work out how drugs interplay with your genes and how gene variations play a role in this, we should be able to develop better drugs and more individualised medicines, treatments that are tailored to your particular problem and that suit your particular gene problem. We should also be able to increase things like the safety of drugs. It's not much point giving something that's not going to be useful if you've got a different sort of gene problem. So we can work out things like adverse drug events, things like efficacy, which one's going to be suitable for you. In fact, there are many adverse drug reactions, and quite a number of people actually die each year. And I think the 2 million hospitalizations is strictly in the US, and this was about 5 years ago. So it's something that we know is a problem, and if we can identify beforehand, we may be able to reduce the number of hospitalizations. We may also be able to make better use of existing drugs or drugs that haven't been found to have a good use. But ultimately what we're after here is trying to tailor drugs to an individual's problem. What I want to do is just give you a bit of an overview of some of the research we've been doing on one particular disease, and that's on migraine. And we've been doing pharmacogenetic studies on migraine with the aim of personalising treatments, that is, trying to develop better diagnostics and more efficacious treatments. So migraine is an incredibly common disorder. It affects about 12% of the population in Australia. It's actually about 18% of women are affected and 6% of men. So 3 times more women than men are affected by migraine. And it doesn't really matter what population you look at. These results here are from different sorts of populations from different countries around the world. And any country you look at has a very high incidence of migraine, so there's about 10 to 12%, sometimes even higher, but in every case there's about 3 times more females than males affected by it. In terms of when people develop their migraine, there's actually a big difference in the genders as well. You'll find that in boys, quite often it can be a fairly young age of onset, right down to about the age of 4 or 5. Whereas in females, even though some can get it quite young, the majority of females develop their migraine round about puberty. So round about their teenage years is when they first start getting it, and it tends to be very bad in their 20s and 30s and may drop off a bit later. So there's a big difference in females developing migraine round about puberty. Migraine also shows what's called comorbidity, meaning that people who suffer from migraine also tend to suffer from other disorders, in particular cardiovascular disease and in particular stroke, also epilepsy, even diabetes, and a number of psychiatric disorders, in particular clinical depression. So it could indicate that there are perhaps shared genetic factors that maybe play a role in both of these conditions. In terms of what are the types of migraine, there are 2 main types. One's called migraine with and the other without aura, but both of them are caused by obviously having migraine. That's the common feature of them, but the migraine itself or the head pain can vary enormously in the intensity and the frequency and the duration. But it's not just head pain, it's associated with nausea and vomiting, photophobia and phonophobia, meaning you can't stand bright lights and loud noises. It also has enormous variation in how long it lasts. It can last from about 4 hours up to several days. All the features I just mentioned are common to the 2 sorts of migraine, but those who suffer from migraine with aura, in addition, have a number of strange neurological disturbances just before the head pain. And it can be things like flashing lights, waving line, even to tunnel vision or even complete loss of vision. So visual disturbances, speech defects, they can even have numbness and weakness and even paralysis of part of the body and even coma. So these aura neurological symptoms are associated with the migraine. And what we've been trying to do for a number of years is try to identify the genes that cause this disorder. The disorder shows what's called strong familial aggregation. That means it runs through families. You tend to find that somebody who has it has a mother or a grandmother or a father or a brother, somebody else who suffers from the disorder. There's been a number of twin studies that support a significant genetic component in migraine, and it's probably around about 60 to 65% genetic, with some known environmental triggers. And what we believe this means is that there's probably a genetic background to migraine or genes that play a role in it, and then there are some environmental triggers that can bring on an attack in those that are predisposed to it. And the triggers can be certain types of food, red wine, chocolate, pressure changes in the weather, sometimes even whether you are or you aren't on a holiday, stress levels, things like that. So there are triggers but it only occurs in those with the genetic background. In terms of how many genes are involved in migraine, when we first started working, there was absolutely no idea how many genes there were, and we thought perhaps there would be one. We started working on this in the 1990s. We now know that there's multiple genes involved in migraine, and some of these have been identified. It's a particular subtype of migraine with aura called familial hemiplegic migraine, and 3 different genes have already been identified as having mutations in it that definitely cause the migraine. One of them is a gene that controls calcium level in the brain, another one is one that controls ATP levels, and the third one is one that controls sodium levels. And the last one is actually a gene that has mutations that cause migraine in some families and cause epilepsy in other families. So when I mentioned that there could be shared genetic factors, well, there's a gene that actually causes both. And similarly, the calcium channel gene that I mentioned can cause migraine, mutations can cause it, but it can also cause a number of other neurological disorders. So what can we do with this information? There's 3 genes that we know are involved in causing migraine. Well, what we can do now is we can diagnose those people. And currently our lab actually does that. We have diagnostic tests for 6 different neurogenetic diseases, including these 3 here, and we can provide back clinicians with the information, does your patient have a mutation in this gene or not? If they have a mutation, you can then diagnose them with that particular type of disorder, and there are some treatments that work very well if you know that that's what they have and it's not something else. So that's starting to personalise your medicine. The information from your genes will tell you if you have that particular gene mutation, and then that information provides information to the neurologist or the clinician who can then decide what sort of treatment would be appropriate for that particular person. But in actual fact, even though this is very useful and it's something that is ongoing and we currently will continue to do, it's actually, these are pretty rare forms of migraine. These are not the common types of migraine. So we're particularly interested in trying to find the genes that relate to the more common types of migraine. And the way to actually do that is to try and investigate genes involved in this disorder in 2 ways. So one, you can use families and look at genes that run through the family. And secondly, you can look at gene variation in cases, people who have the disorder, and controls, people who don't have the disorder. And we've been using both approaches. We've been using large families, and this is an example of a migraine family. The people who are coloured in are the people who have the disorder. The people who aren't coloured in don't have the disorder. The squares are the males and the females are the circle. And you can see that just looking at that family there, it does tend to occur in most generations, so it tends to be inherited in what we call a dominant way. Not in every case, but in most cases. And you can see that you can actually get quite large families with multiple affected members of the family. And that's what we've been doing. We've been investigating large families that suffer from the disorder, and we try and work out what genes correlate with the disorder through the family. And using that approach, we've been able to map a number of genes to particular chromosomal regions. And our studies have implicated a region on the X chromosome and also another region on chromosome 1. Also, why am I sticking up a picture of a boat? I'm sticking up a picture of a boat because we have another very large family that we're also investigating for migraine, and that's a family from Norfolk Island. And Norfolk Island is an island that's located about a 2-hour flight east of Australia or east of Brisbane. It's an island that's derived, 80% of the population of the island are derived from a very famous historical event, and that's the mutiny on the Bounty. So basically there was a mutiny in the 1700s. A number of people from that mutiny, and in fact 9 Bounty mutineers, took a number of Tahitian women, 12 of them with them, and got rid of everybody they didn't like, and they took the boat. And they took the boat to a very isolated island called Pitcairn Island. They then proceeded to have children, and many generations later, they were rediscovered in Queen Victoria's time. And what she did was pardon the offspring of the founders and decided to, they were starting to run out of food and water quite seriously by then, but she decided to let them have their own island and go somewhere else. So she put them on a boat, most of them, and she sent them to Norfolk Island. And Norfolk was an uninhabited island at the time. It was an island that I said is a territory of Australia and located reasonably close to Australia, but it's very isolated. For a geneticist, this is a pretty exciting little island because the majority of the people on the island fit within a single family. They have very strong family groupings, well-documented family histories. They're very proud of their history. And in fact, we've been able to fit 80% of the island's population into a single family, 12-generational family, with about 6,300 individuals in that family. So remember the picture I showed you before of the squares and the circles? Well, this one's just a little more complex. It's so big that we can't fit it all, and in fact, if I was to draw it out like the square and the circle one, it's actually 42 pages long. So this is a kind of shorthand way of showing that everybody ties up together in that family. And for us, it's a very large family that we're currently using to investigate migraine genes. We've been able to map a region to a particular chromosome, and we're currently looking at genes in that chromosome. So that's the second way that you investigate genes. If you can map or define which chromosomes involved, you can then start to investigate the candidate genes in that region. And so to do that, we use migraine cases and controls. And we have about 1,000 migraine cases, individuals who've donated their DNA to us so that we can investigate their genes. And we have about 1,000 controls that we then investigate specific genes in the implicated regions. Now, I'm not going to go through years and years of work. I'm just going to cut to the chase and give you a couple of interesting snippets of those studies. So we've investigated many different genes, different classes of genes, genes that relate to neurotransmitters such as dopamine genes, serotonin genes. We've investigated genes on specific regions that we've implicated, such as a chromosome 19 gene. But I'm just going to focus today on two areas. One is hormone genes, and the other would be vascular genes. So we actually investigated hormone receptor genes because of the big difference in gender. 3 times more females suffer from it than males. So we thought, well, perhaps there's a role for hormones here, particularly since females develop it during the puberty years more often. So we investigated variants in a number of genes that relate to various hormones, and we found variations in 2 genes, the estrogen receptor gene and the progesterone receptor gene. Both of them had specific variations that showed up much more frequently in people who suffer from the disorder compared to people who don't. And in fact, if you had both of those, the estrogen and the progesterone receptor one, you were even at a higher increased risk. So basically what we're doing now is investigating people, in particular a menstrual migraine population, to see whether that obviously has a big hormonal role and to see whether that plays a significant role in that. And then the last gene that I want to mention is a gene that relates to vascular function. There's a lot of overlap between migraine and stroke, so things like loss of vision, numbness, speech problems, thank you. Headache, differences in blood flow. These are things that you can find in people who are suffering from stroke, but also people who are suffering from migraine. So we decided to investigate a number of genes that might play a role in both. And in one in particular we investigated, it's called the methylene tetrahydrofolate reductase gene, MTHFR. This gene has a variation in it. It's been implicated as if you have this variation, it's a functional variation, so it's a mutation. The mutation actually causes a change in the enzyme and it results in a high level of something called homocysteine. And high levels of homocysteine are associated with increased cardiovascular disease and stroke. We investigated it in migraine and we showed that the same mutation occurs much more often in those who suffer from migraine, and in particular migraine with aura, the one that has the weird visual disturbances, etc., that you find in stroke too. So, this particular gene has a mutation and results in high levels of homocysteine. More importantly, in the 1960s it was shown that you could modify the level of homocysteine by increasing folate and vitamin B in your diet. So what we decided to do was to target that particular gene mutation and see if it had an effect on the homocysteine levels in migraineurs. So we did a double-blind placebo-controlled trial on people with and without the mutation. And to cut a long story short, when we gave them folate and vitamin B, we could show in their blood that it increased the folate and vitamin B in their blood in those on the treatment. We also could show it also dropped down the homocysteine levels, even in those who had the mutation. So we could drop down homocysteine down to normal levels. But more importantly, we showed that those who were on the treatment, it actually had a significant effect on their migraine. So it actually, almost there, one slide. So it reduced their migraine disability, it reduced their pain, and it reduced their severity. So we were able to show that you can use genetic information to tailor a new treatment that actually works very simply. We've just done a bigger trial that supported this. And I suppose all I want to say here is this means that genetics information can be used to come up with new treatments, and I think there's going to be a lot more of this. This is personalised medicine using genetic information. Thank you.